Thank you for the nice introduction. And as I was actually expecting, well, the actually there was another speaker before me, right? So, but anyway, I'm going to talk about the bacteria protein deimmunization, and as the using the molecular dynamics and as the protein design techniques to reduce immunogenicity and at the same time to enhance the catalytic activity, enzymatic activity. So, well, I mean, to use the bacteria, I mean, especially the opportunistic, the pathogen as a the therapeutic agent. It's not, I mean, it's not the crazy idea of the mad scientist. It's actually one of the earliest treatments for the cancer therapy. And as well, this person is from the Johns Hopkins University, so he used a bacteria, so to treat the cancer. And the comparing the conventional the chemotherapy and as the bacteria therapy, I mean there was the definite advantage of using the bacteria therapy because so that it replicates, and so it's a living thing. So the chemotherapy is the only small portion of the chemical compounds goes into the tumor, so you have to treat it several times. But as the bacteria therapy, it replicates, and for some reason, and so we don't really know exactly how it works, but it just likes going to the tumor site, and it just kills the tumor. So it's just a magical thing. But so we still don't know exactly what is the mode of mechanism and the action mechanism of the bacteria therapy. So there are a number of the bacteria strains to treat the cancer in clinical trials and the preclinical trials. So including the strains like the VMP and the V1R and SAM. All those things, I mean, they use the opportunistic the pathogens. I mean, they want to kill the cell. So it's basically pathogens to treat the cancer. So it's ongoing. So we don't know the results. Nothing has been fully approved by the FDA yet, but it's promising. But one major bottleneck is, this is all about this conference, is the immunogenicity. So if you use the opportunistic pathogen as the therapeutic carrier, then you expect that it's bacteria. So it's not human. So it should induce the immune responses. So it's one of the, it's not one of them, one of the major bottlenecks. It's the major bottleneck. So fortunately, mostly is as mentioned earlier, so we want to use the opportunistic pathogens to kill the tumor cells. So the many strains that we use, the Salmonella strains to treat the cancer. So, and it has been quite well studied. So which part is immunogenic, which part is toxic, and so all those kind of things. So they just don't use the bacteria itself. It is engineered, highly engineered the protein. And as we already know that which proteins are immunogenic, and so those are the two well-known immunogenic parts. So the one is the toxin, and the other one is a phosphatase. It's the shortly called the PhoN. And those two are the most immunodominant antigens across the Salmonella cerevisiae. It's pretty well known. And in order to treat those immunogenicity issues, so the simplest solution is just to get rid of it. If the toxin is not, for example, toxin. It is toxin, so we just get rid of the gene from we can just knock it out, and it's not going to be made, and we are just safe to use to avoid the immunogenicity. But sometimes if the gene is essential for the survival of the bacteria, then we cannot just simply remove it. And unfortunately, PhoN is such the protein. So in general, PhoN is a non-specific phosphatase, and there are lots of genes that does the job. So in general, in a normal conditions, if you just get rid of it, then the bacteria still survives. But if the environment as a vitamin B6, the depleted environment, and as unfortunately, the there are many tumor microenvironments. The vitamin B6, they are just our cells, and they uptake a lot of the vitamins as well, to replicate themselves. So in many the tumor microenvironments, vitamin B6 is just depleted. So our purpose of using this therapeutic bacteria is to kill the tumor, but when it goes to tumor, then vitamin B6 is gone, so it cannot survive. So the PhoN6, although it's immunogenic, we can't just get rid of it. So we need alternative strategy. So as I said, it is essential. But we can't just get rid of it because it cannot survive in certain tumor microenvironments. So the alternative strategy is to make the protein less immunogenic. The Edmund already mentioned about the deimmunization techniques by protein design. So this is the purpose of the protein deimmunization. So we want to keep that gene, and at the same time, so we want to reduce its immunogenicity. But here, we made another kick, so that we enhanced the enzymatic activity to make the protein even more potent. So in order to understand how the enzymatic activity happens, then we need to understand the dynamics of the protein. So we performed the molecular dynamics of the enzymes. The ligand is present and is not present. So we found that the certain positions, which is highlighted in green color, it's when the ligand is not present, and if it is present, then the dynamics changes quite a lot. So if it's not present, then it moves a little bit. And then if it is present, ligand is there, then it's solid. So we understand that the dynamics of the particular position is important in the enzymatic activity. So our goal is to reduce the immunogenicity, and especially it's quite close to that gate, the position. So our purpose is to reduce immunogenicity, but at the same time to enhance the dynamics. Optimally enhance the dynamics of that gate. So in order to enhance the enzymatic activity at the same time. So, this is a kind of like a technical details, but basically we use the Rosetta and it's the IDB, the consensus method, to reduce the immunogenicity. So basically we pick the mutable amino acids using the in silico screening of MHC binding. Then as they use the Rosetta the design techniques, to find the structurally the good amino acids. Then as we also perform the molecular dynamics to see the gate movement as the optimally the produced. So here's the one part, one important part that we would like to emphasize is that we didn't introduce mutation in general. So when you want to make the mutations, then you make mutations on the surface because it's just safe. But here is just the proof of concept, and so we want to disrupt the protein itself. So we want to disrupt that immunogenic part so that it also makes the movement on the gate of the enzymatic activity side. So we intentionally made mutations in the core part of, buried regions of the epitope. So that we just took the more challenging the protein design techniques. And so this is just the MHC epitope map. And so we first, after running the Rosetta design, then as we took certain the protein sequences which have the lowest Rosetta energy, and so at the same time we produced like the 4,000 the sequences. Then sometimes it just hits the same sequences all the same time. So we just pick like the two criteria as like the most populated ones, and at the same time the lowest energy conformations. And as we checked those like designed the sequences, is it like immunogenic or not? Then using the helping from the help of the proimmune. Then so we performed their assays, MHC assays. Then as we found that as our designed sequences are like they're not. It doesn't really do the antigen presentation, so it doesn't do like the MHC binding. So now it's time to look at the molecular dynamics. Their dynamics, and so we saw that obviously we intentionally made the mutations in the buried regions, so this immunogenic part is moving quite a lot. But at the same time, we want to make like these regions, gate regions of the enzyme, gate regions of the enzymes, moving, need to move, so to enhance, to control the enzymatic activity. And so we saw that as the certain regions, the changes, and as we performed the RMSF, the root mean squared, the fluctuation, the patterns, and as we used like the PCA analysis, and as we split it, and as so just to see the patterns, and as we picked like the 5 variants, and the variant 1 is the molecular dynamics pattern is similar to wild type, and as there are like the 3 groups, and as the 2 of them are like somewhere in the middle, and the 2 of them they move quite a lot. So we have like 3 groups of the dynamic groups, and as we found that this is one example as the variant 2, especially, and as you can see, this ligand goes away. So basically, the molecular dynamics shows that this particular enzyme is not really suitable. It's expected and predicted to be less enzymatic, the activity. So this is particularly this variant 2. The videos, the video clips that you saw is from the molecular dynamics results of these 2. So we expect that these particular groups, which moves too much, then as it shows like less. They reduce the enzymatic activity, and as expected, this the molecular dynamics pattern similar to wild type, it shows more or less the same as the wild type, the enzymatic activity. But in the middle, the 4 and 5, somewhere in the middle, the optimal the dynamic the groups, they shows up to 3 to 4 times the enhanced the enzymatic activity. So yeah. So our conclusion is the dynamics actually matters, and so if you move, if you make the particular gate, the enzyme gate, move too much, then as you break down the enzymatic activity. But if you make optimally and somewhere and the moderate the movement, then so that you can make the enzyme as a more active than the wild type. So the conclusion is here is like the immunogenicity is that we would like to make the enzyme the less immunogenic, but at the same time, so that we would like to make the enzyme more active by changing its dynamics, and so that we show that as proof of concept. So we reduce the immunogenicity, and at the same time, so we made the enzymes that are more active. So all this work was performed by my students. She's present here, and so we have the poster number 2. So if you have any technical details to ask, then so you can come to me or my student here. And it was funded by the NRF and it's the C&Cure, which is the company that uses the bacteria as like the therapeutic carrier. So yeah, thank you. Yeah. These are my daughters. Yeah.
Thank you for the nice talk, and certainly an exciting area to be able to make a real difference through deimmunisation. I had a technical question about the use of the assay with the different peptides, because it looked like you'd used some different peptide lengths, and I think it showed it was essentially the same sequence in the 2 peptides that gave you the signal, so like would be confirmatory. Yeah, on this slide, great. And I think in some reports, like different lengths of peptides can give different outcomes. Yeah. So probably nice to see that you got the same, but I wonder what your thinking was in designing this experiment.
Yeah, so the length of peptides was already given because of the original papers. They found it from the LCMSMS, and then as they read the peptide that is already loaded onto the particular actually I think the B10101. But anyway, so that we knew that is the particular length, but the proimmune they recommended exactly the same, saying that is but probably so we could try like different lengths, like 15 months, all the different lengths. So that's why we saw that as but in any case, our design sequences are all like so not present in the MHC. And fortunately, as you said, is the like longer one and shorter ones. I mean, the core part. I think that the core part is still conserved, and it shows like the same binding affinity. So yeah.
And so as well, this was the IEDB algorithms that you used to design the alternatives.
Yes. So we used the different algorithms. So initially we used the IDB consensus method, and as later we also tested the MHPAN 4.3. But when we initiated this work, the net MHPAN, the newest version wasn't there. So we just to check it, just to double check it, and so we also used the PropRed. Because this is just the technical details, but here is like in the Rosetta design, so we use the mover is called MHC epitope energy, and it's a default version is actually PropRed, which is very old one, but it's we saw that it's correlated with the IDB and it's that MHPAN, fortunately in this case. So yeah,
that's a lovely case study. Thank you for sharing it.
Thank you for your talk. I have a question. How many substitutions have you made in?
It's actually I can't really recall
in the successful.
So I think it's 10 mutations. 10, 8. How many mutations have we introduced? Huh? 5 to 7 mutations in, and it is a very short fragment. Like I think it's the 19-mer, but it's the, we introduced like the, yeah, quite a lot.
Yeah.
Yeah.
Okay. Thanks.
Thank you, Professor. I have a question. What is the strategy you use to deimmunize your protein sequence? I mean, which amino acid you will use to mutant, and what is what amino acid sequence you will mutate to by randomly mutation or specific substitution?
So the initially we ran the blast. So to find it's a relatives. So I think it's the net from learning from the natural sequences would be safer. So that we ran a blast, and as we took out the sequences relative to this our protein. Then as it shows like each position, so it shows like naturally occurring the mutations. Then as we run the NetImage pan or like IDB consensus, which reduces the binding the activity of binding activity. So initially it's like not all 20, but it's from the blast is we took some the listed amino acid. Then so we filter it out again by running the MHC binding the predictors. Then as we run again on the structure. Then as the Rosetta and as the other at the time the protein and PNN was not there, but that's why we used the Rosetta design. But anyways, the structurally after filtering out the several steps, then as the structurally allowed the amino acids are the selected using the Rosetta. So that's why we came up with the only handful number of the sequences, like the 5 sequences. So.